All right. Hello, everyone. Can you hear me up there? It's okay? Thank you. Well, it looks like most of you were here yesterday, so it looks like you guys are again stuck with my presentation at the end of a very long day, and I'm really sorry about that. So I hope that you still can get something useful out of this. But basically today, as promised, I was going to focus on our tool MetScape, and I will try to point out the things in the workflow that were mentioned in Barbara and Chuck's talk, and essentially explain to you guys what we're doing to try and extend that workflow so you can go from a tool like CoolMap to MetScape and so on and so forth. But I'm going to start with the basics, and this is the slide that I showed yesterday. And I briefly explained the workflow, so I'm just going to tell you what I'm going to focus on today. So today I'm going to focus primarily on metabolomics data. I don't know how and when it happened, but we have been developing this tool for a number of years, and it got to the point where the workflow is rather complicated, or it can be complicated if you want it to be. And so my talk tomorrow on data integration is going to be focused on that part of the workflow. I'll show you what you can do with gene expression data. But today we will just stick with metabolomics. And again, all the credits for the development of this tool goes to our outstanding developers, Bill, who is here and who is going to give you a hands-on demo, and Terry, who has been involved in the development of this tool from the very beginning, Tim Hall and Glenn Tarsier, who worked on MetScape previously. So everything you like about this tool, if you try it, all credits go to them. Everything you don't like were my bad ideas, so you can blame me. So anyway, I just want to briefly remind you that we have a database behind this tool that contains human metabolic networks data, and in addition to that, we are using, you will see when I show you the tool, you will see options for inputting rat and mouse data, and that applies only to gene data. So we're using homologene to map rat and mouse genes to their human homologs, but the metabolic network is essentially human. So there are some issues with using that, for example, for Drosophila data. So as you might expect, not every Drosophila gene will have a human homolog. I'm using Drosophila as an example, okay? And these are the basic objects in our database and the basic type of nodes that you will see in the tool. And so if you want to use the tool, it's useful to have a data set that you can actually load, and I think it always goes better. And so I'm going to use this really simple data set for the demo. It comes from the study done by our colleagues at UC Davis, where they looked at plasma samples from patients with pancreatic cancer and healthy controls. And so it was a large untargeted study, and it had a subset of identified metabolites, and some of these metabolites were significant by simple t-test, and there is a fold change. So as you can see, it's basically a 3-column file. And so in this case, we're using KEGG ID to identify the compounds, but you can, as I will show you, you can actually use compound names. The second column is p-value. In this case, since we had 2 groups, we used simple t-test, as I said. And fold change or log fold change can be loaded into the tool. And if you don't have fold change, or if you don't want to look at that for whatever reason, fewer visualization features will be available to you. So basically, this is optional, okay? And with this, I think we're about to find out if demo gods are smiling at me today. So I'm going to actually try and show you. So this is, I already opened the Cytoscape. This is a very basic screen that you're going to see when you open Cytoscape. And so you see across the top, there's a menu with a row of buttons. And MetScape, as we said before, is an app for Cytoscape, or plugin or app. And it lives under this application menu, and I already have it installed. And those of you who actually are signed up for a hands-on session on Thursday, Bill will show you in great detail how to install it, and how to start using the tool. So I'm just going to go directly to build network. So I selected that from MetScape menu, build network button right here. And what it does, it opens this MetScape panel, which is a basic sort of, MetScape tool that you can use to, as I said, select an organism. And in our case, we're going to be using human. And this button here allows you to select an experimental file. I won't do it right now. Bill will show it to you, but I will just say that it's possible to enter the compounds and genes likewise by directly typing the names of the compounds. But in this case, you are limited to just, you can build a metabolic network, but it won't have any experimental data. We kind of reasoned that if you want to enter experimental data, you probably will want to load that from a file. Nobody wants to type that, right? So I'm just going to select a file. And so this is the basic menu. This is another place where you can choose an organism. And if you are loading metabolite data, it's really not that important. So it probably won't complain. But if you select the wrong organism and try, for example, if you select human and try to load rat data, genes, it will complain and you will get an error, just so you know. Okay, so I'm going to select a file, and that is on my desktop. MetScape demo folder. And so I am going to select this. This is the very same file that I had on my slides. That's what I'm entering here. Now, notice that there are these two drop downs here. So basically what happens is, as you saw in my file, I had columns corresponding to p-value and fold change. And so this allows you to map columns from a file. So conceivably, you can have p-values and fold changes for a number of different groups, or you can have other columns, as I will show you. And so this really allows you to map these. And that is where you can set the significance threshold. And I'll show you where we will see that in the data. And so I'm just going to leave it at default. So all fold changes are going to be significant, and the threshold for significance for p-value is going to be 0.05. So we're just going to do this very quick study. Okay, so we're not going to, that's where you would enter gene data, which I will be talking about tomorrow, and Bill will show you. And tomorrow I will also talk about that part, concepts in great detail. But for now, we're just going to say, okay, this is a very important screen. You can have a number of compounds in the file that you're trying to load, obviously. And so not all the elements that you have in your file, especially if you have compound names, are necessarily going to be mapped into our database. And this is where you get the information about the elements in your file, in this case compounds, that were not mapped, okay? The reason it's important is because typically you want to know that. If there were some significant metabolites and you're not going to see them in your network, you're going to wonder where they are and so on and so forth. So the only way to preserve that information is to click that save button, and it will just save it as a text file, okay? This way you will have access to it. Otherwise, if I click OK, you would have to reload the data to bring that dialog back. So I think it's important to point that out. Now, to the next part of the network panel. It was mentioned several times that we have different types of network graphs, and this basically describes the type of nodes you're going to see in the network. Again, I'm going to start with this more detailed version for now because we're going to build a relatively small network, and I will not talk about these options right now, but we'll get back to that later. So I'm just going to click Build Network, and that should be relatively fast because we only had a few compounds. That shouldn't take too long. And in fact, we're done. So here's our network. So now this, as you can see, it was black before. Now it got populated, this window. So things that you can do here, and if you're not familiar with Cytoscape, you can, the first thing that you want to do, what are those small things there? You, of course, want to zoom in, right? So now I am zoomed in, and some of those things kind of went outside my screen. There are a couple of different ways to do that. In this case, one way to navigate your network screen is to move this rectangle here, which hopefully you can see what I'm doing, right? So now I can zoom on one of these sort of, clusters, if you will, okay? And so I'm going to put it in the center of my screen, and that's where you can start reading the names and so on and so forth. So first big red blob that we see here is arachidonic acid. And this is a very busy network. So lots of sizes and lots of shapes and colors, sorry. And so if you want to get more details about that, what you can do is you can go and show the legend, which will explain what different types of shapes and nodes you see here. And so these hexagons are the compounds, right? And so one thing that you immediately notice is that some compound nodes are this salmon pink color, and our arachidonic acid here is red. And what it means is simply that this was our input compound, okay? In this case, I'm going to zoom out for a second. We have a relatively small network. Probably wouldn't be too hard to find our input compounds, but the bigger your network gets, the more difficult it is, obviously. So that's just a way to keep track of your input compounds. Everything else, this pink-colored compounds and other information, has been pulled out of our database, which, remember, has information about how all this these compounds are linked by metabolic reactions and enzymes and genes and so on and so forth. And so if we zoom back in here, then and follow the legend for a minute. So the diamond-shaped gray nodes are reactions, and I don't have any green nodes in this view, but here's one. By the way, if you are on a PC and using a mouse with a middle mouse button, you can sort of accomplish the same thing. You can move your network graph. You can pan, basically, okay? And so the green nodes here, when they're selected, they are no longer green, they're yellow, but the green rectangular-shaped nodes are enzymes, and connected to them are genes that encode them, right? So in this case, there are a couple of genes here. And if you want, in general, if you want to get any more information about any of the elements in MetScape network, what you can do is simply double-click on a node. In this case, I double-clicked on a gene node, and I'm going to just resize it a little bit so we have a little bit more room here, right? And so here's the information. Here's some additional information about this particular gene. So we can see the not only entry gene ID and gene symbol, we can see the description. So in fact, this is a cyclooxygenase and some subcellular location where this gene is expressed and links to some databases. Similarly, I can close that for a minute. Similarly, you can get more information on other types of nodes. So for example, for compound, you will get names, synonyms, different types of IDs if we have them in the database, links to various databases. Obviously, they will be slightly different from gene databases, links to different databases, and you will have a structure for the compound. So I'm going to close that again for the moment. So this is sort of, your sort of, high-level view of the MetScape network. I want to draw your attention. I'm going to close the legend for a second, and I'm going to draw your attention to a lower part of the screen here, okay? This is what we call pathway filter. So we made some decisions very early on when we were developing MetScape. We decided that we're not going to restrict the size of the network artificially, okay? So we're going to allow all the elements, genes, compounds, reactions that are at least one step away, will allow them to be incorporated into network, which means that if you have your input is 100 100 compounds and maybe 2,000 genes, that is going to be a pretty sizable network, okay? And so we decided that it's okay to have huge hairball, basically, because what we're going to do instead, we're going to build in a number of different filters, okay? And so what we are doing is, so one of these filters is right here. We call it a pathway filter. And so essentially what it is, it's a list of pathways in which the elements in this network are involved. And so if I can, well, not surprisingly, we looked at arachidonic acid, and of course it's part of arachidonic acid metabolism. And so you can select one or more of these, in fact. So if I look at bile acid biosynthesis, and zoom in here and move it a little bit so to bring it into focus, then I'm going to see the compounds and associated reactions, enzymes, and genes that are involved in bile acid metabolism. So you can select one or more of these pathways. So let me zoom out so you can actually see what I'm doing here. So, if I select more pathways, more elements get selected, and so on and so forth. Okay? So this is a sort of, very first sort of, look at the tool and very brief overview of it, overview of the tool. And now I want to go back for a minute, and I want to continue with the presentation to sort of explain a few other features that I would like to talk about today. I mentioned to you that you can have a more complex input file, and you can put more data into MetScape, okay? So what I'm showing you, the first part of that file is exactly the same as before, right? It has the same three columns: compound ID, this one, this example comes from PUFA data, but it's not important here. The point is that I have p-values in this column. It says t-test PUFA versus carbohydrate. And the second column, which you cannot read very well, says fold change. Okay, so these are this part is exactly the same as before. But look, here I have two different conditions now. Okay, so I have baseline, and I have time point 1 for PUFA, and time point 2 for PUFA data. And similarly, I have these time points for carbohydrate data, okay? And these are normalized measurements, by the way, okay? So, and what you can do is you can actually take this file and you can put it into MetScape, okay? And I'm going to show you how to do that, except that I'm going to use a different data set, a slightly different data set that also comes from Chuck's lab. And in this case, they looked at the effect of prolonged, basically metabolic changes that occur in response to exposure of beta cells, cultured cells, to high fatty acids or high glucose, okay? And this is a sort of heat map style display, if you will, of different metabolites that are divided into different pathways and how they're changing with exposure to glucose, I believe, in this case. Okay, in this example, and this is just sort of a heat map style display. Okay, so that is the data set that I'm actually going to put into MetScape. Okay, and I have screenshots of that just in case I don't know how to, just in case something wouldn't work. But what I'm going to do next, I'm basically, since it's a larger data set, it takes a little bit, I mean, it doesn't take too long to load it, but it's enough to sort of, make me uncomfortable by its loading. So I'm going to do it as a cooking show. Now imagine that, you put this in the oven and, an hour later, voila. Okay, well, it's not an hour later, it's just, maybe 40 seconds later, but these are, so it's a pretty large data set. There are something in the order of 90 metabolites that we loaded here, okay? And so they have been loaded, and that results in this network, which is resembling a hairball, I think, pretty much, right? And so as we discussed before, all these red compounds are actually input compounds. And so one thing that I would like to sort of, show you is how I will be using these other extra columns in the data, okay? And what it allows us to really sort of understand about this particular data set, and I believe that Chuck will be coming back to that tomorrow in his presentation. Okay, so as we discussed before, I have my pathway filter here. That's this tab here, and I'm just going to point out one more thing. So I think they may be already sorted alphabetically, but If they're not, or if you wish to resort them, you can always do that. And in this case, it's pretty complicated, but with this glucose exposure, one of the things that would like to you probably would want to look at is you probably would want to look at something like TCA cycle, right? So I'm just going to find the TCA cycle here, and all the nodes that belong to TCA cycle pathway are selected. And then all this sort of, what I said about the filter, that basically, is embodied here in this sort of, short workflow that I'm showing you. So what I'm doing now, what I've done, I just created a subnetwork for this subset of nodes with one click of a mouse, essentially, okay? So now I'm looking at TCA cycle compounds, okay? So now, first of all, this is not, for those of you who know your biochemistry, this is not a complete TCA cycle. This is just a subset of nodes that belong to TCA cycle in my network. If you do want to build a complete TCA cycle network, the way to do it would be right here on MetScape panel. You can select, instead of selecting instead of using input compounds, you can go to this button, and there will be a drop-down list of pathways, okay? But we'll just stick with this because it gives me sort of, an ability to show you another interesting feature of MetScape. So I'm going to zoom in on this network, and suppose I would also like to include more compounds in this pathway, or in this network that I have on the screen. How would I do that? Suppose I want to expand it. And I'm also interested because I know, maybe, or I'm wondering, have we measured also the levels of acetate, aspartate, and maybe I would like to look at that. So what I'm going to do, I'm going to right-click on oxaloacetate node here and go to, that brings up the menu, and I'm going to go to MetScape menu, and I'm going to select to expand, and I'm going to expand it an existing network. And let's see what happens. Okay, so we will have to zoom in again. And unfortunately, what happens when you do that is the layout has changed a little bit. But the node that we expanded, oxaloacetate, is now, you can see that the name of that compound is now bold. And so we got some extra compounds and extra nodes and extra edges. And these new expansion edges are blue. And by the way, what I haven't mentioned, but you probably noticed that some of these arrows are double-headed, and some of these are, some of these are directional. And so wherever we have the information about the reaction, whether the reaction is reversible or non-reversible, we represent that with arrows, okay? And so you can see that glutamate and aspartate just got added to this network, and judging by the, so these are expansion compounds, that's why they're pink, but this green border really shows that they were in our experimental data set. And again, Bill is going to go through all these features in a lot more details on Thursday. But for now, what I would like to do, I would like to zoom out a little bit. And so suppose this is a network that we would like to explore. And now what I would like to do, I would like to see how these metabolites that we've measured, that are here right now on the screen, how they're changing over time, okay? And the way to do that, we have a feature to do that. And we have to go back to MetScape menu and select this animation feature. And what that allows you to do is to actually configure the layout where we can paste the columns and measurements the way it makes sense to us. So in this case, I'm going to have 3 data series, and my data series are going to be control, PUFA, oh, I'm sorry, in this case, it's fatty acids, and this is high glucose, and the first column is going to be at the baseline, and then I'm going to move the subsequent times here. You can do it in any order, so that's how you sort of set it up for your animation. And if you make a mistake, by the way, you can always sort of clear it with the restart button and start over, but let's see. I don't think I messed anything up, so I don't have to do that. Okay. So once I have set up my data grid, all I have to do is to click on this button and say build animation. Okay, and notice that my screen has actually changed, right? So instead of one network that I have now 3 different networks. Okay, so it's the same network essentially, but I'm showing the measurements for control right in the middle, and fatty acids and glucose networks right here. And similar to what Chuck and Barbara showed in CoolMap, I have an ability to actually change. What I'm going to do, I'm going to set it to green-red, just so it resembles the heat maps that you guys have seen earlier. And so if you have any sort of, outlier values, you can sort of, move these sliders, and I'll just mention it. And with this, so essentially, I have this sort of network. So now I can look at this changes dynamically over time, okay? The other thing I would like to mention that, suppose I want to kind of zoom in on a particular place in my network and control, and for some reason I want to look at closer at malate and citrate and so on, right? And so what I would like then, probably, it makes sense to realign other networks so they would be in exactly the same position if I want to do that, okay? So if I zoom out again, just so you can see more nodes, for example, I can again realign, and it will be all aligned. And then, so basically, this is going to go through my time points and show me how these metabolites are changing, okay? And there's a number of other features and tools that we're developing that I would like to get to. So I am going to leave it to Chuck to sort of, talk about the biological implications of this particular study and how he's using this to analyze the data and use it in his work. So I'm going to stop the animation, and so if I don't want to play it as a movie, what I can do is I can just sort of, click at one time point at a time. And so notice the color changes and size changes, as those correspond to the fold changes that, the concentration changes that were in our input file, okay? So that's basically what I wanted to show you with this part of the demo. And so now I'm just going to exit, and it's going to ask me if I'm sure, and then it sort of, will return me to this sort of view. Now, all of the data that you have here can be exported in the tab delimited format, and Cytoscape itself provides the ability to export the images that you, to the networks that you see as graphics, as images. If you ever get to the point where you have a network that you would like to publish, feel free to send me an email, because I spend endless amount of hours trying to figure out how to get it to 600 dpi to satisfy most journals requirements, and I'd be happy to share with you, but I'm not going to do that right now. So for now, I'm just going to go back to the PowerPoint. And by the way, the slides have been uploaded to the website, and I do have the screenshots of all the steps that we took. So you will be able to kind of retrace that, creating subnetwork and expanding network and so on and so forth, right? So now I hope we convinced you, or at least sort of, piqued your interest in terms of maybe trying out this tool. So I think it would be only fair to tell you about the limitations of this tool, and likewise any other tool that attempts to do pathway mapping type of thing. Okay, and so I had that slide yesterday, but we went through that very quickly. But basically the point is that the canonical pathways that we're using here are not going to cover, likely not going to cover all the metabolites that you've measured, unless you have done a targeted study where you measured amino acids and, for example, TCA cycle intermediates or something like that, in which case they're all going to be here, but then you probably can sort of draw it by hand, right? For the untargeted study, even if you're doing study in human, remember we have human metabolic networks here. But you're also likely to see nutritional compounds, various compounds of environmental origin, compounds that come from microbiome. Okay, and these would be bacterial metabolites that are not going to be part of human metabolic networks. That's one of the big limitations, right? And so going back to sort of, Barbara described very well sort of, this overall sort of workflows and processes. So what this is, in essence, we're using prior knowledge to kind of extend our understanding, explore our data, and put our data in the context of prior knowledge, right? I mean, and this is a great thing to do, but in a way it's limiting. What about novel pathways? So the argument that I often hear, how am I going to discover something new if I'm sort of, throwing away the metabolites that are not mapped into these pathways, and so on and so forth. Okay, so, and this is something that many people are trying to work with, and so I'm just going to briefly mention a tool that we have developed here that we call Metab2Mesh. Okay, and what that is so, it has been mentioned previously when Sub gave his overview yesterday. And so in this tool, we started with a much broader database than just a database of human metabolic pathways. We started with PubChem database, which I talked about yesterday. It's huge, right? And then what we did, we downloaded the entire PubMed, and we do it periodically to update our tool. And so National Library of Medicine basically annotates all published articles with medical subject headings, or MeSH terms. So if you go to PubMed database and look at an abstract of a publication, you can click a link there, and it will show you MeSH terms, medical subject headings. Okay, and they also generate a file of various substances that are mentioned in published literature. And what we did, we basically merged these two data sets together to link PubChem compounds to published literature. Okay, and we did it in a completely automated fashion because it's a huge data set. And but believe me, there are a lot of sort of very spurious links right there. So for example, we found that, there is a compound that is called DNA. I don't remember what it stands for, but it's not deoxyribonucleic acid. And as you can imagine, that gets a lot of PubMed hits, right? And so essentially to eliminate the non-significant matches, we used Fisher's exact test, and we took only the most statistically significant associations between compounds and articles. And that resulted in this data set, and these are my colleagues, Maureen Sartor, my collaborator, and Alex Aid and Zach Wright, who were involved in developing this tool. And so this is a web interface. So essentially what you can do, you can either search by MeSH terms, and it will give you a list of compounds. In this case, I searched by cyclic AMP. And this is a list of, I'm sorry, in this case, I searched by a compound, cyclic AMP, and it gives me a list of MeSH terms that are associated with this, and I can filter it by different categories. In this case, I'm filtering it by diseases, right? So I can find out that it has been linked to different cancers and so on and so forth. And there's a link here, which I'm not going to show you, but Bill, again, will demo this tool. He will not demo this tool, but briefly show you this interface, I think, on Thursday, right? So you can actually go to the articles that substantiate this connection. So now what I want to mention next is that, well, since we already know how to work with Cytoscape, we kind of decided that it would be useful, in addition to this web interface, where essentially you can search it one compound at a time, which may not be terribly useful. We thought, well, wouldn't it be great if we could annotate the metabolic networks in Cytoscape with MetDisease terms, right? So What I'm showing you here is a snapshot of our other tool, which we also published. And I don't have a reference here, and I apologize for that. I think I made that slide before the paper got published, but it was published in Bioinformatics this year. And we call it MetDisease. So essentially, it's sort of a small addition to, it's an app for Cytoscape. And you can have the differences. I mean, you can certainly do it with MetScape network, but essentially you can have any network you want as long as your compounds have either PubChem IDs, KEGG IDs, or synonyms that we can recognize. And what you can do then is you can essentially find related mesh terms, and they will pop up in another panel down here. And so what I'm showing here is essentially, when you click on one of these, the respective compounds in the network get selected. So in this case, I'm selecting the compounds that have been linked through published literature to brain neoplasms. And if I right-click and go to MetDisease menu, then it will take me to the actual PubMed and give me the abstracts of the papers that substantiate this connection, okay? And so the very important point here is that it's a standalone tool. It will work with MetScape, but it's a standalone tool, which means that you can put any network in here. And in fact, networks that I'm displaying here, the nodes, you notice that they're colored differently from the MetScape network because it is a different network. This comes from a publication by a German group. And in this case, the nodes in the network represent metabolites, and the edges actually represent correlations. So essentially what it is, if I took the matrix that Barbara and Chuck showed you, if I took a correlation matrix where I would just, calculated the correlation coefficients between compounds and set certain threshold for significance. So, for example, I would take only most significant correlations. Then the compounds would become nodes in this network, and the correlation coefficients would become edges. Except that in this particular case, this published this sort of published set of metabolites, what they did, instead of calculating simple Pearson correlation, they did something that is called partial correlation, which essentially allows you to take into account many additional variables. So it just does not calculate correlation between two metabolites, but it also takes into account the influence of other factors, basically, okay? And as long as you can sort of, express them numerically, it can be influence of other metabolites, or it can be influence of other, factors in your study. And in fact, this seems to be a very sort of, popular idea if you look at the metabolomics, sort of, informatics data analysis literature. It seems to be a very sort of, popular notion right now. And so what I would like to show you is a little preview of what we're working on right now. This is the next version of MetScape, which should be available hopefully by the end of summer. If we're not ready to release it, at least it should be available in beta version. And if you're interested, you would be welcome to contact us and try it. But what I'm showing you now is a slightly different panel. It's also a MetScape panel, but notice the difference here. It says correlation based. So what this instead of what this program, what this module expects is essentially a list of correlations rather than the list of metabolites and fold changes or concentrations or something like that. Okay, and so there are various options here, various things that you can do. So I'm just sort of, highlighting it to go back to the basic MetScape panel that I showed you before. You would select a different one from this drop down, and this is where you would load the file. Now, I would have to stop here and to say that, my question, personally would be, okay, well, correlations, I mean, you're all fancy, you're talking about this. I mean, George mentioned that as well, you're talking about this partial correlations. So this is something that we felt would be better done outside of Cytoscape. And so what we're working on is essentially an algorithm that George and his graduate student are involved in, have been involved in developing for quite some time now. And so we are going to basically, release it with the companion program that you can use if you want to generate this type of correlation matrix from your data. Now, you don't have to use it. If you have your favorite algorithm for doing that, you're welcome to use that. And the format, again is very simple. And that will be available when we're ready to release that. But anyway, the screen that you would see once you have selected and loaded your file, in this case, so the metabolites or features are identified by name. But since we're no longer limiting it to metabolic pathways, there is no, your compound is not going to be discarded just because it's not found in our database, which means that potentially you can put all the unknown features in that load your unknown features as well, which is a serious sort of, expansion of what we did before. Right, but in this case, in this example, we are showing a couple of different compounds that were not found, but they will be loaded nonetheless. And so the majority of these were found. And I didn't show you, this is pretty similar to what MetScape does right now. If you enter a compound name, Bill will show this to you. But, if you, if it gives you this drop down and you want a different compound, you can see what else has been found in the database, okay? And that's sort of, once you are satisfied with your input, you can click OK. And then this panel gets populated with the data and various cutoffs are available to you. So I'm showing you a pretty small network here. So notice different color nodes. These are colored the same salmon color as they are in MetScape, basically, but there are a couple of compounds that were highlighted in red that were not mapped, even though there were known features. This is a targeted data set, by the way, so there were no unknown features. But the point of this is to show you that your unknown features potentially can be these white nodes here, and all the edges are correlations. So I didn't filter it by anything in this case, or Bill didn't filter it because he did all the screen captures for that. But so notice also that edges now have different color, and the color in this case represents positive and negative correlations. Positive are red, negative are blue, and they have different weight. The strongest correlations are, these heavier, thicker lines, and you can sort of do a number of different adjustments here with these sliders, and you can restrict things to a certain level. You have various options to put information about, if you have any information about groups in your data, such as, for example, if you have a lipidomics study and you know what lipid classes you have in your data, you can sort of also put that information. And you may want to look at correlations between different classes of lipids, because what we know is if we have measured a group of free fatty acids, chances are that they will be strongly correlated, and you will not see other correlations that you might be looking for in your data. So we want to be able to say, okay, give me between group correlations only, and that's all of that is embodied here, okay? And so if I were to, if it would be a live demo, since it's an early version, we just didn't feel that we were ready to give a live demo of that. And but I would be adjusting the sliders and right here. And when I adjust that, and it keeps on the most significant correlations, these edges that were thicker. So that's basically how it works. And so if you remember, I previously had a fully connected network. And so now I have two outliers that are not connected to anything, just because the correlations of these two to the rest of the network were not sufficiently strong to meet my cutoff criteria. That's kind of the visualization that's the visualization in the next MetScape modules that we're working on right now. And I think we're getting close to the point where we can, let people play with it and test it. So, what I also want to say is that we're always very happy to let people test our tools, and if you want to work with us, and if you want to give us feedback, or if you don't want to work with us and just want to give us feedback, that's cool too. And we really appreciate it, and that really helped us a lot to implement many new nice features. Okay, and so I think I'm just sort of going to conclude that to give credit to the people who are involved in this work. So we have our small team who's working in correlation network. Samantha is no longer a graduate student. He actually graduated, and so he worked with George and Chuck and Bill are involved in that work. And so the main sort of, future directions and applications of what we're doing, where we see that is, I briefly mentioned to you mapping lipid pathways, and I pointed out the unknown compounds. And in fact, we do know that metabolites are not changing independently with each other. Some metabolites tend to be strongly correlated. For example, branched chain amino acids, or many others, right? And so we hope that this tools and some features that we would like to add to it would actually speed up the prioritization of unknown features. Okay, I mean, note that I'm intentionally sort of avoiding the word identification. We won't be able to identify them, but what we can do, we can prioritize them, and we can narrow down the search space, if you will. Okay. And the other project that is going on, we are looking pretty hard at doing displaying metabolic networks for other organisms. I mentioned Drosophila, and it's not by accident. We're actually looking at Drosophila because we got a lot of requests on that. And so we have a pilot project for Drosophila, but if it works, it can easily be expanded to other biopsych databases, which I mentioned yesterday. Okay, so we're also working on putting in additional compound identifiers because, among other things, our colleagues at San Diego DRCC decided for metabolomics data repository to use PubChem IDs, and we want our tools to be compatible with what they develop, so it's a high priority for us. And the other project is, George described in some more detail enrichment analysis. And I'm going to mention some of the things that we're doing with it in my talk tomorrow, mostly with regard to gene expression. But I have a wonderful collaborator, Marin Sartor, who is an assistant professor in our department, and her expertise is in enrichment analysis. And we're building another new tool, which we call Concept Metab, but more about enrichment analysis tomorrow. So I think I'll stop here, and I'll be happy to take any questions. Yes.
So the network view that you show, is it possible to map it to a regular metabolic pathway like in a textbook so it's more reader friendly?
Yeah, it's a very good question. I think, let me see if I understand. For example, you would want to have a more sort of traditional display of a metabolic pathway, like KEG, for example.
Like the TCA cycle. I want to map it.
You want to see it as a sort of TCA cycle. Well, it's a very good question. So this is really a network tool, right? So what I I showed you some tools yesterday, including the omics viewer from Peter Karp's group. That's how they do things, okay? So we felt that the main strength of our tool is in being able to link different pathways into a single network. We toyed with the idea of sort of laying out these nodes in a sort of, more sort of, KEG-like pathway structure. And it didn't go very far because our networks are messy because, what we didn't want to do, we did not want to put one metabolite on the map several times, right? So if you have your acetyl-CoA here and here, that sort of, breaks our networks. So our next networks built the way that, each metabolite is unique. It occurs there only once. So what we do instead, we do various filtering. So I think the short answer is no, you cannot lay them out exactly like this. But somebody has developed, I saw a publication from somebody in Japan who is not associated with KEGG. I think this person developed his own plugin for Cytoscape that actually lays out KEGG pathways in a sort of, this nicer view. And so the beauty of working with Cytoscape actually is that there is a very large community of developers who are developing these various plugins. So it may be possible to actually use their tool to lay out MetScape networks. We looked at that at one point, and I think it can be done.
Okay.
So
yeah, I think right now the best out there is probably the omics. It's the nicest tool right there. So can you export the data to omics right now?
Can you export the data
from your data to the omics tool, omics to visualize?
From one tool to another? Well, I don't think you need to export it from our tool to their tool.
Okay.
And the reason for that is if you have, most tools will take tab delimited file, and your data initially probably is laid in columns and rows and so on and so forth. So there's, you can export data from our tool into a file and put it into their tool, but you probably are better off starting with your data set as it comes in, basically.
Okay.
Okay? Yes.
Automatic process, or is it an automatic process, or is it something that has to wait for next version of MetScape?
No, unfortunately, it's not an automatic. No, what needs to happen in order for this to this change to appear, I think what you would have to basically reanalyze the data, but that would require us updating the database, which is something that we're actually doing right now because there is a newer version of human metabolic network. And I think I mentioned that yesterday. So once we update that, it will be a new database. But then I think what you would need to do is to re-import the data. Unfortunately, this sort of backtracking is kind of a difficult thing to do. So unfortunately, the answer to that is no. It would be nice. I agree with you. Any other questions? Thank you.